Okay, good. Hello, everyone. So this is going to be a demo about iCn3D. And the first part I'm going to present, and then you can ask me questions in the second session. So iCn3D is a web-based 3D viewer to replace previous CN3D at NCBI. And there are many ways you can use iCn3D. The first one is you can use in the web browser, and here shows the link. And you can use in Jupyter Notebook, and you can write, I have some example scripts, but you can write Python or Node.js script to analyze your structures in command line. And you can view, use iCn3D, view 3D structures in virtual reality, augmented reality, and you can also print your 3D models. And the source code is in GitHub, and it's also in NPM. So, so each slide, I think I have a share link. If you can open the slide, then you can click and to show exactly the same view I show here. So first, in order to use iCn3D, you need to load a 3D structure. And there are two options. One is input your PDB or AlphaFold IDs. And second option is input your PDB file. So once you open iCn3D, you can Google search iCn3D, and then click the file menu. There's retrieve by ID, and you can, this option, you can input either PDB, MNDB, or AlphaFold IDs. And in the open file menu, you can input multiple PDB files. So these are the typical ways to input your 3D structures. And once you load the structure, you can rotate, zoom, or translate, use different, if you use a mouse, then usually it's a left, middle, or right mouse. And there's also some key you can use to do rotation. And if it's a MacBook or phone, you can, use click and drag for the rotate, pinch and spread for zoom, and two finger for the translate, two finger click and drag. Yeah. So iCn3D, similar to CN3D, it synchronizes the 3D, 2D, and 1D sequence. So in the analysis menu, the first one, sequence annotations, show the sequence and plus annotations in tracks. And then the 2D diagram or 2D cartoon is a 2D display as shown in the central panel in the bottom part. And defined set also, it's convenient. You can define your own set or the predefined set for the chains. So this will be also, you can consider as 1D or 2D. Yeah, probably 1D. So once you select in 3D, 2D, or 1D, it will be, the selection will show up across all views. And one thing I want to stress is iCn3D work only on your selection. So once you select something, your style color will only apply to your selection. So by default, all the items are selected. But once you select something, this will become orange and shows selection. So because iCn3D is command-based, and each command will only work on your current set. So you don't need to do the selection for each of your, you do selection once and all the following command will only work on your selection. Sometimes it might be confusing if you accidentally select something and then you suddenly you want to change the color, only apply to a certain region. So I just want to stress that. So you can select in 3D, 2D, or 1D. Select 3D, you go to select menu, select on 3D. Here is the hint. You can click the Alt key and plus click. And in MacBook, it's option, and the control in MacBook, it's command. So you can figure it out. And by default, if you press Alt and click, it will select the residue, but you can change to select an atom or select the chain. And in the sets, it's straightforward. You can select multiple set with the control key. And 2D diagram, you can click the node to select a chain. And in the sequence, you can drag on the sequence to select the sequence. So once you do selection, this toggle will become selection, and the style and the color also change to orange, indicate that now you are only working on your selection. So once you select, for example, a few residues, and you can view selection, then only your selection will be shown, and this will reflect it in the 2D, and your selection will automatically be saved in the sets in most cases. Yeah. So this is view your selection, and then you can change style. These are pretty much straightforward. So schematic, it's a kind of new in iCn3D. Basically, it shows a residue with a name and show the C alpha trace and with the residue name. So it's convenient to see the sequence as well. And the top part shows different display style, and this part shows the parameters for the surface. And then you can also show electron density. And the color is also straightforward. The unique color, you can define just one color for your selection. And the rainbow and spectrum, rainbow from red to violet, and spectrum the opposite. And secondary structure might be convenient. Sometimes you can show the sheet by, typically people show the sheet in yellow, but we use yellow a lot or orange a lot, so you could show in green. And there are other ways to show different color. So for example, you finish all these steps and you want to save your work. And there are two ways to save your work. The first way is to save iCn3D PNG image. When you do save file iCn3D PNG image, it saves two files. One is the image, the other one is HTML, which when you open the HTML file, it will show up like this. You click and it'll show the view. So the HTML file is a way you can, for example, if you have many custom views, you have many HTML files. If you concatenate those HTML file as a source, concatenate the source code, you actually have a gallery of your work. So there will be a gallery of this image in the same page. So iCn3D image is not, iCn3D PNG image is not only an image, it also contains the commands and sometimes even the PDB files. So it can be loaded back to iCn3D. So if your input is the ID, then the PNG image contains a command. If your input is a custom file, there's no ID to retrieve, right? So the PNG image actually contains a PDB file so that when you load it back, it can read the structure. So this is the preferred way to save your work. It not only has your view, but also you can reproduce it by load it back to iCn3D. But if your input is ID, you have a way to just save your work in a link. Go to File, Share Link. So for example, I did something, and all the commands are recorded in the full URL. And this full URL will be shortened using Google Firebase, and then will be a short URL you can share with your colleagues. You can also publish it because the commands and the ID are all recorded in the URL, and this will be lifelong, and we support backward compatibility, so it will always be supported. So by now I just show the basic steps to do to use iCn3D to view your 3D structure and save your work. Those are very basic features. And now I'm going to introduce more features in iCn3D. Basically, they are analysis features. They are more, detailed and yeah. So the first thing is you can show annotations in iCn3D. Go to sequence annotations. There are many sequence-based annotations like conserved domain, claim var, snips, functional size, post-translational modification. And these are based on the sequence. And there are also structure-related annotations like 3D domains, disulfide bonds, interactions, and cross-linkage. So you can choose to select some of them or select everything. And once you select everything, then all the annotations will show up as tracks. So here, the first line is the sequence, and then shows the conserved domain, claim var, snips, and these are the functional sites and post-translational modification and the interactions with other chains. And you can click the detail to view all the sequence. So this is just the summary view. And annotations can be retrieved with both Node.js script or Python script. So the Node.js script, the example is at GitHub page and with the name annotations.js. So there are many kinds of annotations. For example, if you want to retrieve post-translation annotation and it assign a type as 9, so you just type node annotations.js 9. Let me think. You need to somehow specify the PDB ID. I think the PDB ID is specified in the script. Yeah, I think so, because you can modify a script. So you specify the PDB ID in the script and just type this command. You will retrieve the post-translational modification. I think those residue number, and the output is a JSON format. But you can also use a Python script. The Python script works slightly different. Node.js is more powerful. It can access any data in iCn3D. Python script can retrieve all the data from the UI. So basically, there's a URL. You can actually, when you click in the UI, when you click this, it saves the HTML page for this. And this URL can be just replaced in this script. Just replace the URL. You will get the, the output, the HTML page as output. So you just type the command. Again, everything is defined in this Python script. Next, I'm going to talk about the interaction analysis. this is very popular. So if you go to analysis, there's a feature called interaction. And once you click interaction, it will ask you to select what kind of interaction you want to show. So for example, hydrogen bonds, salt bridge, contacts. There are many contacts. Sometimes you want to leave it out. Halogen bonds for the chloride, fluoride, those elements. And pi cation, usually it's between the charged group and the aromatic group. And pi stacking are usually, like, for example, in DNA or in the hydrophobic residues. Those aromatic residues, aromatic rings can stack together. Those are pi stacking. So you can also define the threshold. And then you define the two sets. The first set, for example, you want to get all the ligand, and then second set, you could be the proteins, but it could be your own defined set. And then there are different ways to show the interaction. Here I'm going to show you the 2D interaction network. Basically, one set will be in one line, the other set will be in the other line, and the interactions will show up as lines. So here the different color indicates different interactions. So red is pi cation interactions. So you can clearly see the interaction between two sets. And in 3D, I also show the same color for different interactions, and the dashed line indicate the interaction. Okay, then I'm going to talk about mutation analysis. If you go to analysis, previously I show you interaction, right? Now you go to mutation, and here you specify the PDB or AlphaFold uniprot ID, then the chain, then the residue number you want to do mutation, and the last one will be the residue name of your mutant. So this one, and the E chain of 6M0G, that's the SARS-CoV-2 structure. E chain is spike protein at position 501. I want to mutate it to tyrosine. And then you click, you can output the PDB, or you can show interactions. In this case, N is a wild type, and then mutate to tyrosine at position 501. So it introduced pi cation and pi stacking and lose one hydrogen bonds. So these are the difference, but it also show all the interactions and the common interactions. Here, I just want to show the difference of the interactions. And under each menu, there are 4 icons from the menu. So the view that's alternate. So when you click that one, you will alternate between the wild type and the mutant. Then I'm going to show you how to show the electrostatic potential in iCn3D. And in analysis menu, there's a Delphi potential. So there are 2 ways to show the potential. The typical way is show the potential on the surface. So these are the surface of the protein, and blue indicates it's a positive charge, and red indicates negative potential. So these are the potential on the protein surface. So in the ligand, near the ligand, it's pretty much negative potential. And for the ligand, I show the equipotential map. That means, for example, blue mesh shows positive 75 millivolt. It's a 3 kT equal to 75 millivolt. And the red shows minus 75 millivolt. So you will see the electrostatic interactions between the ligand and the protein. Why there are two ways to show it because, for example, for the blue part, anything above 75 millivolt is just blue. You cannot tell the difference. But when you show the equipotential map, if it's, for example, 150 millivolt, then the mesh will be further away. So that means you will see the difference. Yeah. So yeah, the equipotential map is more accurately represent the potential. Next, I'm going to show how to search similar structures. So there are three ways to search similar structures. One is you want to search similar structures as a complex, PDB complex. For example, 1-HHO, it's there are 4 monomers, so you want to search for the complex, and basically click this, you can go to VAS plus to find similar structure. Actually, the input in this case is not a 1-HHO, it's a SARS-CoV-2 structure. Sorry, I mixed these two. So, and then you can also, once you get to VAS plus page, you can search text or PDB ID in the search button, and then you can, for example, these two spike protein, and you can view in iCn3D. So that's search for the protein complex. If you just want to search for a chain, for example, AlphaFold structure, typically it's just one chain. So if you want to search AlphaFold structure, you can search similar, go to NCBI VAST. So in VAST, if it's a PDB structure, you can input the PDB ID and the chain name, and the VAST similar structure will show up, and you can select certain chain and show in iCn3D. And next, I'm going to talk about the AlphaFold structures. So you have a loaded structure or AlphaFold structure, and then you can, for example, you can load your AlphaFold structure in iCn3D and just click submit, and then the loaded structure will submit to VAS neighbor search, and this is a VAS search page, and you click VAS. There are two steps. One is show a summary, and then you start the VAS neighbor search. And then once you find a neighbor, then you will see the previous one. You can select a few structures and show in iCn3D. And once you find a similar structure, you can, for example, you know they are similar from VAST. You can directly show the alignment in iCn3D. So, for example, I know these chains are similar. I can put them in file align and align them by structure or by sequence or by residue means predefined residue mapping. So we have two ways to align them, either by TM align from John's group from University of Michigan, or you can align with VAST. And it will show the alignment in 3D and also the sequence, multiple sequence aligned by structure in this case. And remember, we can retrieve by ID to load multiple structure or open files, load multiple PDB files, right? You can load them and do the alignment later in iCn3D. We call it realign selection. So here you can realign your structure based on your selection. You can select certain chains. If you don't select anything, then the whole structure will be used. So you can go to multiple chain by structure alignment. And once you click that, it will ask you to select selections. Or if you don't do anything, then everything will be used for the realignment. And then This will be realigned. In this case, the realignment is using TM align, but doesn't matter. It just sometimes TM align works better, for the realignment. And then in realignment, its pairwise alignment will be shown, because in realignment, for example, if you have the SARS-CoV-2 spike protein, if you have multiple antibody, you could use the realign to assemble the spike protein with different antibodies. So they basically it's pairwise. So